Hi, I'm Radha,
And I'm David.
and for our project this summer, we worked on RNA-seq deduplication with UMIs under the guidance of Dr. Barbara Novak.
So, on to the background. Let's first explain what exactly is RNA sequencing. Well, it essentially involves the use of next-generation sequencing to reveal the presence and quantify RNA, such as messenger RNA, long non-coding RNA, and microRNA in any biological sample. One goal for doing all of this is to profile gene expression by identifying genes that are differentially expressed during two or more biological conditions, such as being in space versus being here on the ground on Earth. By undergoing this process, duplicates will eventually be created through the amplification process. But in this context, what exactly are these duplicates? Well, when you sequence complementary DNA, you get multiple short sequences, which are referred to as reads from each sample. Therefore, duplicates are when you get more than one read with the exact same sequence. So to your right, the diagram shows a complementary DNA strand at the very top. The same strand is then cut up into four multiple short sequences of equal length, and when we look at those four resulting sequences at the very bottom, we see that two of those are of the same color, to represent two strands being identical. So with these duplicates, they can be classified into two main groups, either biological or technical. With biological ones, they are considered true duplicates that come from multiple copies of a transcript from the original sample. So essentially that means that these duplicates were already present before any amplification was done to the samples. With technical duplicates, however, they were introduced and created from a technical aspect from either the library prep or sequencing. So in this context, when we mean deduplicating, the goal is to remove technical duplicates.
So UMIs, or unique molecular identifiers, help labs keep track of molecules and remove errors during the library preparation and sequencing steps. Before the PCR amplification step, which is the step where we create a bunch of these technical duplicates, every molecule in a sample is uniquely labeled with a UMI, which is typically a random sequence of oligonucleotides. When we see two identical tags on two identical sequences, we can assume that they are from the same original molecule and are therefore technical duplicates. A finding of two different tags on the same sequence means that the sequences must have come from two different original molecules and are therefore biological duplicates. As of now, there exists no single approach to efficiently removing technical duplicates using UMIs. There are many programs that exist, but they are very time and memory intensive. So through this project, we evaluated the efficiency of two different deduplication methods, which were Picard's marked duplicates and UMI tools dedupe.
Onto the methods used, I first used FGBio's copy UMI from read name to parse through every single read name to copy the UMIs at the very ends to ultimately create a SAM or X tag that Picard could later use. So once that's all said and done, I then inputted those modified files to Picard's marked duplicates so it could eventually do its thing and remove any duplicates found based on those molecular tags that were created earlier. So this seems pretty simple and straightforward. Radha will now go over just how UMI tools fared during this process.
So for UMI tools dedupe, the command requires a sorted and indexed alignment file. So I had to do that first with the samtools sort and index commands. Then I was able to run UMI tools dedupe. After that, I had to run the deduplicated file through samtools collate, which would sort the file by name. And then lastly, run it through the UMI tools prepare for rsem command in order for it to be compatible with rsem, which is the next step of the GeneLab data processing pipeline. So for the results, here is a table showing the duplication rates of a few samples without UMIs, and then with our tools, UMI tools, and Picard. One thing to note is how the duplication rates without UMIs is significantly higher than the duplication rates of UMI tools and Picard. This is likely because the duplication rate without UMIs was measured using only the alignment position. This means that this duplication rate probably includes both biological and technical duplicates. UMI tools and Picard, on the other hand, use their UMIs along with the alignment position in order to flag duplicates. This means that any fragment that got aligned to the same location could now be differentiated between biological and technical using their UMIs. So these two tools having significantly lower duplication rates is exactly what we'd want to see, considering that we want to leave the biological duplicates alone and only remove the technical ones.
So to ultimately conclude, our current results suggest that both tools could work in deduplicating GeneLab RNA sequencing data, but one important thing to note is that the differences in duplication rates could be attributed to just how strict the two tools are in determining if two UMIs are duplicates, where Picard requires an exact match, whereas UMI tools allows for at least one mismatch.
So for our future plans, we would like to run differential gene expression analysis on the deduplicated samples in order to see how deduplication might affect those results. We also want to look more into why UMI tools and Picard duplication rates were so different from each other. And we also need to optimize these tools for running it on the GeneLab cluster, because currently they were very time and resource intensive, and we need to work on making them more efficient if we want to be able to run it consistently. And then if applicable, in the end, we would like to update the RNA-seq pipeline in order to include deduplication.
So as of now, with this slide here, here's the current RNA-seq pipeline. And with this slide here, this is where our work of UMI deduplication would go if this step were to be implemented. So before we end off, we gotta give thanks to where credit is due. Funding for this project was provided by the NASA BMSIS YSP, and we couldn't have done this project without the extensive help from Dr. Barbara Novak, Dr. Amanda Saravia Butler, Alexis Torres, and everyone from the NASA GeneLab data processing team.
Thank you.
Thank you.